Great, Dr. Njoroge.
Also, good afternoon, guys. So we'll just get right down to it. So number one, which one of the following organic bases of nucleotides is a double-ringed purine? Personally, nimesahau hizo purines and pyrimidines. So if someone can just share them, so that's those of us who are writing down and write them down. Someone out there?
Okay, I think I can do that.
Yeah, please.
So pyrimidines the examples are cytosine, uracil, and thiamine. And then for the purines, it's adenine and guanine. So which will make the answer B?
B, yeah, but B because guanine is also double ringed. Sindio, uracil is not, uracil and thiamine are not double ringed. Sindio?
Yeah.
So. Purine, sorry. So, yeah. I was I was reading, sorry. So number two, the building blocks of triglycerides are, I think that one is D, glycerol and fatty acids. Amino acids are the building blocks for proteins, and monosaccharides and disaccharides play the building blocks for carbohydrates. Sindio. So, which of the following of these statements about osmosis is true? Osmosis always involves a membrane that allows water and all solids to diffuse through. That is, I think that's wrong because we're talking about a semi-permeable, meaning it's only permeable to some things and not permeable to others, semi-permeable. So the greater the solid concentration, the smaller the osmotic pressure of a solution. I think that's also wrong. I think an increase in solute concentration increases the osmotic pressure and therefore almost attracting water from the other side to come to the side which is highly concentrated with solids. Osmosis occurs because of hydrostatic pressure outside the cell. I think that's also wrong. Osmosis, I think it has more to do with the osmotic pressure other than the hydrostatic pressure. As we saw the other time, we were saying hydrostatic pressure is the pressure which is caused by the movement of blood through, within the blood vessels. So that's also wrong. And also think of it this way: a lower hydrostatic pressure leads to your blood or whatever being more stagnant and therefore providing more time for osmosis to take place. So you'd want a lower hydrostatic pressure, yani less movement for osmosis to occur. And then lastly, the greater the osmotic pressure of a solution, the greater the tendency for water to move into the solution, just as I've said. So a higher osmotic pressure is present when we have more solutes within our solution. So when we say something is hypertonic, it means it has a greater osmotic pressure and it has more solids within it. And therefore, there's a greater tendency for, remember, the water wants to move from an area where it is highly concentrated. It's almost like diffusion. When you think of it this way, like osmosis is just like diffusion, but now we're talking about water. So the water wants to move from an area it's highly concentrated to an area where it's less concentrated. So the water wants to come from a place where it's highly diluted to an area where the solute is highly concentrated. So the answer is D for number 3. We can go to the next question.
Sorry, Willie, you said you go for the answer for?
The answer is D, dog.
Yeah.
Yeah, okay. Lysis of a cell is expected to occur when this, okay. Divya is saying she doesn't feel like it. Divya, please switch on, if you can't just switch on your mic and me and you can have a small conversation.
Diva is a clown.
Oh,
Yeah.
oh. sorry. So that's a great picture actually. Divya, if you can share the picture to the group. So sorry for that. So lysis of a cell is expected to occur when the cell is placed in which of the following solutions? Higher. So we've just spoken about osmosis, and you said water likes to move from where it's less concentrated to where it's more concentrated. Sindio? So basically, we would want the cell to be more concentrated than the water for the water to want to come into the cell. So the answer in number 4 is a hypotonic. Yes, Black Panther, thank you. Yeah, so yeah, just remember osmosis is like diffusion, but now we're only talking about water and we are involving a semi-permeable membrane. Higher, a cell that synthesizes large amounts of proteins. See, I'm a clown, sorry. So a cell that synthesizes large amounts of protein for use outside the cell has a cell that synthesizes large amounts of protein. Protein come from ribosomes. For use outside the cell has go for rough endoplasmic reticulum, because the rough endoplasmic reticulums are used for transporting the proteins. So we see that they're rough because they contain ribosomes on them. Higher. What is the percentage of body water in an average adult male? Body water, I think they usually say about 60 to 70%. So I'd go with the answer C. So let's go to number 7.
Will, I think just for number 6.
Uh-huh.
Number 1 to 5 are all those concepts we've gone through them before. But for number 6 specifically, perhaps Shrina can advise. I've missed a few sessions, but have we gone through the fluids, ICF, interstitial, the percentages? There are usually those percentages between ICF.
Didn't do the percentages.
We didn't do.
We didn't do the percentages, but I think we did talk something about ICF and ECF, but I can't recall.
Yeah, we did talk about it in terms of electrolytes when we said
Yes.
ICF, potassium, yeah, such things.
Yeah, but we didn't do the
Other percentages. Okay.
Yeah, maybe we can do it now.
Kindly. So yeah, anyone who remembers those percentages between ICF, ECF, how in short, fluid distribution in the body in terms of percentages can share just on the group, or they can just say it here.
So I can I've seen like a picture here. Just cause to avoid wasting time. So intracellular, it's saying the intracellular fluid makes up for 2/3 of your water present, of the fluid present in the body, which is about 67%. And then the extracellular fluid makes up the rest, which is 1/3. And then from that, 1/3 is about 33. Sindio? 33.3. Yeah. So, and then the interstitial fluid, interstitial fluid is about 26%. So I'm not sure 26% of the total body
think
fluid.
it's from the 33 because the ECF now, when you like the subdivision of ECF is plasma and the extravascular fluid.
Yeah.
Then extravascular fluid is now
And
the
also the cerebrospinal, but that's less than 1%.
yeah.
Yeah, but the 26% I'm saying is from the total, not
Or
so
not
it's
from the 33.
not from the 33.
Okay.
Yeah. I don't know if anyone has those fractions. At least I've shared for the intracellular and the extracellular. So intracellular is the most with 2/3, and then extracellular 1/3. And then now maybe if someone has interstitial and intravascular or blood plasma, maybe they can share.
I've shared on the group
Mhm.
the percentages and even the volume. So yeah, I think that suffices as a reference.
Super. Let me just see. So that's great. So I can go to the next question, or do you? Yeah, but I can just read out the picture. So basically, the total from what you've shared, the total body fluid is 42 liters, and that's 100%. And it makes up 60% of our body weight. Intracellular fluid is 28. Let's even forget about the volume because that's just cramming. We can just focus on the percentages, and then depending on what, depending on what you get. So we've said total body fluid, that's our 100%. As we said, intracellular fluid, intracellular, so within the cells, that's 67 or 2/3, while extracellular is 33%. And then from there, interstitial is 80% of the ECF, while plasma is 20% of the ECF. I think that's more than enough. We can remember. So just remember 80-20. And the funny thing about this is like what you expect is not what you get, because someone would expect your extracellular fluid to be the most, but now that's less than your intracellular. And then you'd expect, you'd expect your plasma to be more than your interstitial fluid, but that's not really the case. So that's an interesting way of remembering it. Anyway, so let me go to the next question. Which of these factors does not affect the body metabolic rate? Muscular exertion, age, emotional state, and environmental temperature. Personally, I would go with choice D, because in itself, due to homeostasis, our body temperature is always maintained at the same, around the same place. But for muscular exertion, oh no, I think I'd go with choice A, because when we talk about the BMR, we are talking about the body's metabolic rate at rest. So I think age, emotional state, and environmental temperature would affect, but BMR, we are talking about rest, so I'd go with choice A. What do you guys think?
I'm not sure, but I think I'd go for D.
Yeah, even me, I was thinking D, and then I just thought about BMR. And then also remember, like when we talk about the thyroid, it usually, the thyroid plays a very major role in metabolism, like metabolism functions. And remember, like people who have hypothyroidism, they usually feel cold. So I think just the matter of temperature in itself is usually integral when we're talking about metabolic rate. And then just the thing of like BMR, we're talking about the body at rest. So are we really talking about muscular exertion during rest? I don't think so. Because it's not like the metabolic rate at that time, just the body's metabolic rate. Oh no, I'm talking about basal metabolic rate. Sorry. I'm not sure. What do you guys think?
Okay, for environmental temperature, it's the
Uh-huh.
temperature outside the body. So when someone usually feels cold, they tend to burn more calories. It's a way of the body to increase the heat. So you burn more calories when it's cold so that you can give heat to the body. I think that's how I thought
Yeah,
of
yeah.
environmental
True.
temperature. So temperature is very important in that
Yeah.
it affects the body's metabolic rate.
Yeah. So what do you think? What do you think is the answer?
Okay, I don't know if emotion, okay. I don't know. I feel like all the answers are correct, but they're looking for the least
Yeah,
answer
approximate.
here. So I don't know which
Ah.
is the least answer. I will have gone with emotional state, but I don't know.
Okay, yeah, but Shrina has shared a cheat sheet on the group, and it says age, because your metabolic rate slows when it becomes slower when you're older. Body size, the bigger body size, the more calories you burn. Outside temperatures, muscle mass, the greater your muscle mass, the more calories you burn. Movement and hormones. So the only one we're not seeing is emotional state, though I think you can tell how emotional state can play a role somehow, because you're under more stress, under more hizo mashida tu ndio hizo zimeanza kuja. That's what I'm not very sure what, because the only one we're not seeing from the cheat sheet.
But Willie,
Yeah.
from the choices,
Uh-huh.
they've said the amount of muscle that you have, not the amount of muscular exertion.
But they've also
You
said
get-
movement from the cheat sheet. The cheat sheet is like there's muscular exertion, there's muscle mass and movement. So, which muscle exertion and movement are kind of one and the same.
But when you're burning calories, are you burning calories from your muscles? I don't think so.
Okay. I mean, so let's. When were you burning calories from?
Something else was sent on the group, essentially making all answers correct.
Yeah, I've just seen the. Yeah, I think we can put a breather on that one, but maybe tunaweza chagua one answer so that if we ever find this question, we all choose the same thing and complain together later. Yeah. Yeah, but
So what answer are we going with?
I think it's somewhere, I was saying, I thought bodies, because if it was basal metabolic rate, then the answer would be muscular exertion, because now basal means at rest. So I think that's where I'd gone, nearly gone me confused. But body's metabolic rate.
Why would you say that that basal will not be affected by muscle?
Because when you're saying basal, you're talking about the body's metabolic rate at rest. See, when you're saying basal, it's at rest. So it's always the same, even like it's always the same person's body when it's at rest. So you're never considering when the person is active. Am I making sense?
Yeah. But in this case, if you were to compare with someone with less muscle mass, will make a difference.
But this is muscular exertion. So like movement, unajua, not muscle mass, like movement, yani how much they've exerted their muscles. Like if they've run 1,000 kilometers, their basal metabolic rate would still be the same because we're only talking about that rate when it's at rest, not necessarily when they're running right now. Am I making sense?
I think me I would go with C. I think emotional state is least likely.
There's a cheat sheet we've shared on the group where who were just saying increased emotional state will like they'll have a higher metabolic rate because the heart is beating faster, ndio hiyo thyroid gland imeanza imeanza kutolewa hivyo and that's just the thyroid hormone, and that's just associated with, then now think about all the effects of the thyroid hormone once it's released. I think there'll be a lot of metabolism going on when someone has a heightened emotional state. But anyway, we can put a lead in that and just say that answer doesn't necessarily have a question. An answer. That question. So number 8. Which one of the following substances is not found in the urine of a healthy adult female? I think that's glucose. Yeah, and just remember, all glucose is reabsorbed in the kidneys, so and that's why if you find, like, if there's glucose in the kidney, then that's where you expect someone to have UTIs, because then that glucose promotes the growth of these UTI infections, and that's why drugs like SGL2, LT2, are usually associated with UTIs, because then you have increased glucose in your urine, evo. So we expect creatine to be excreted via our kidneys, so that one will be present. Urea, obviously, and sodium. I think those are self-explanatory. So the process by which the curvature of the lens is increased in the eye is called, that's accommodation. Thank you, Rami. Yeah, I think it's lacrimation is basically just crying. Focusing and transduction, I'm not very sure what those are. But accommodation, if you remember that experiment some of us did in high school with the lens, and there's a sort of pinhole and light passing through. Yeah, the good old days. So we can go to the next question, Shrina. Number 9B.
For transduction,
Please.
transduction is just now like that pathway of like when you, like in the dark or the light, like what the photoreceptors do, and then that optic nerve, and just that's just phototransduction or transduction.
So the movement of like the images through our brain too,
Yeah, but mostly with like light, either like in a dark, in like a dim light, or like in like bright light.
Okay.
So you have like the roads and the cones.
Because I think Hiral has mentioned that, and we've never ever revised the phototransduction cascade. Can we look at that real quick? The phototransduction. So how do we see?
Okay, so like a general overview. So we have photoreceptors like rods and cones. So the rods are for the dim or a dark, like dim light, and
Hey, Hiral, are you still with us? Is it me who can't be heard? It's so crazy, you can't even tell.
I think she got cut off.
Okay. Sarafina, do you have any idea, Ama, can you share with us the visual photo transduction cascade?
Sorry, I was kicked out. Can you guys hear me now?
Yeah, loud and clear. Please go ahead.
Okay. Yeah, so I was saying we have photoreceptors like cones and rods. The cones are for the dim light, and the cones are for phototopic vision. So when light hits a compound in these photoreceptors called 11-cis retinal, it converts it to all trans retinal, and that's when now the phototransduction starts now, where there's like a whole thing. So basically, all these signals will go and they will reach now the optic nerve, and then now from the optic nerve it will go to the optic the chiasm, something like that. I think it's better to get a picture, but yeah, from the optic nerve and then it goes to the occipital cortex. But I think the main thing is what these photoreceptors do, and the main molecule that is involved is the retinal. Yeah, but let me find a picture and I can send on the group.
Yeah, so even when you find the picture, I remember that was one of the first areas where we really introduced our G-coupled receptors. So maybe you can eventually also share something about them and at what point do we find them? Yeah, so do I give you a bit of time as we move ahead, or what do you think? Is that fine? And then you can just share the picture on the group.
Yeah, I'll just share it on the group.
Yeah, though if there's anyone who wants to say something, please just feel free. Just come out and just don't even raise your hand, just say whatever you want to say. So number 10, we can go down, Shrina. So the answer of number 9 is B. And as Hiral has said, so lacrimation, just crying, accommodation, curvature of the lens, and then transduction is, you're basically transducing this message through the optic nerve till the optic centers in the brain. So number 10, which of these factors does not affect cardiac output? Higher heart disease, pregnancy, sleep, exercise. Rami says exercise. Personally, I think obviously heart disease, that's pretty clear. I think I'd go with C personally, because I think for pregnancy you'd need a higher cardiac output because of the child. During exercise, we also have a higher cardiac output because remember, our sympathetic nervous system is now at play, and we have increased adrenaline and therefore an increased cardiac output. During heart disease, depending on what heart disease it is, we are likely to have an increased or decreased cardiac output. And I think, so basically sleep. I think during sleep, we have a normal cardiac output. So I'd go with C. Higher, sour. So ovulation is characterized by one of the following: rise in body temperature, a decrease in progesterone secretion, a fall in luteinizing hormone secretion, a decrease in the body level, of estrogens. I think during ovulation, just generally from my thoughts, we have an increase in progesterone and estrogen. Remember, progesterone is there to facilitate the thickening of the uterine muscles, and estrogen, plays a sort of role in promoting the maturation of our ovum. We have a fall in the luteinizing hormone, which then rises later on in the menstrual cycle. So personally, I think I'd go with choice A, a rise in body temperature. Though, can someone please tell me what they think? Question 10, why did we settle for C? Sam, please come out and just share what you think. Feel free.
I don't know if you guys can hear me.
So first tell us what you think what you would have settled for.
Okay. Okay. Before settling for something, you guys can hear me, right?
Yeah, perfect. You're loud and clear. You're doing well.
Because I was wondering, during sleep, the heart rate usually decreases, and we calculate cardiac output as heart rate times the stroke volume.
Yeah.
So if the heart rate is reduced, cardiac output is going to reduce. Right?
Yeah.
Then that means doesn't really qualify.
Sasa,
But I think the heart rate reduces to like a very small percent compared to the other choices where it will increase cardiac output or decrease cardiac output to quite like a higher percentage.
Yeah.
Sorry?
Yes, like the significant changes in the other
Yeah.
Yeah.
Okay, thank you.
So do you agree?
Yeah, I agree.
Situache nyuma. Sawa. Ama, yeah.
Fuck. Oh my goodness.
Sorry about that.
Language.
I hope everything is okay. So, number 12. A major inhibitory substance in the brain is, I think that should be, it will go down. It's glue. Oh yeah, it's GABA. So GABA is the major inhibitory neurotransmitter present in the brain, while the major excitatory neurotransmitter is glycine. Is it glycine or what?
Yes.
So, so we can go to the next question, unless someone has a question.
See, we can just recall the different types of neurotransmitters and their examples for all of them.
Okay, perfect. So yeah, someone can go ahead. Let me take it. So just someone share one. Excitatory glutamate.
Yeah, it's GABA and glycine that are inhibitory.
Sorry about that. So thank you. Yeah, the major excitatory is glutamate. Okay. Ayastano has shared a vital picture. So which states the excitatory are glutamate, aspartate, and nitric oxide. The inhibitory are serotonin, dopamine, GABA, and glycine, while acetylcholine and norepinephrine serve as both inhibitory and excitatory. So, are you satisfied with that, Shrina? Do you want us to add anything else?
Yeah, okay.
So we can go to the next question.
I think Joy is bringing up the previous question. On the chat box.
Oh, so Joy says, I thought ovulation results in an increase in basal body temperature, plus A occurs when progesterone is low and when estrogen is high.
But to answer that, for progesterone raises the body temperature by about 0.5 to 0.9 degrees Celsius. So ovulation is characterized by a rise in progesterone secretion, hence the rise in body temperature. It's not estrogen that raises the body temp, or rather, at least that's what I know.
Gaius, I think it's estrogen that increases. Progesterone only increases after ovulation, because you see, after the egg has been released, you get that corpus luteum thing, which now secretes progesterone. So that increases your progesterone. But during ovulation, it's estrogen and LH that have increased.
Okay, let me just send a diagram that shows during ovulation, the estrogen concentration is usually going down and the progesterone concentration is the one going up.
Just look at the group Fatma sent.
Okay. So ovulation is at the line.
Yeah, it's yeah, exactly. That's day 14, around day 14 when ovulation occurs.
So when we say estrogen secretion increases, wouldn't that be kind of not correct? Because, okay, there's that diagram. There's this one from, this other one I'm sending is from the book of physiology. Because I guess it's more of a perspective of the diagram.
Okay, just send, we see.
Okay. But meanwhile, can we call, so it's estrogen that raises the body temperature, not progesterone?
I think so, but I know during ovulation body temperature increases. I'm not sure because of what, but I know that there's rise in body temperature during ovulation.
Excuse me. The rise in body temperature is usually due to progesterone, not estrogen. It's due to progesterone. And so it usually occurs, in fact, it does not occur on the same day of ovulation. The rise in body temperature occurs at least like 3 or 4 days, around day 18 there of the cycle. So immediately after ovulation, then after some 3 or so days is when we experience the rise in temperature. It does not occur per se on the day of ovulation.
So then what would be the answer in this question?
I'm not really sure of the answer, but just from the knowledge that I've read generally, that is the perspective that I get. But still, you may have to go with rise in body temperature.
So basically from the diagram we've seen LH is up during that period, and also our progesterone rises just after. Yeah, I think A is the best answer that we can give, because basically we're just talking about the characteristics around, it's not like when a lady says she thinks she might be ovulating, the first thing you go is check if she has a fever. So personally, I'd go with A, but thanks for all the added information. So basically we said progesterone works on the hypothalamus as from the group and leading to increase in basal body temperature. We've said LH and estrogen are the first to go up. Remember, we want to form the corpus luteum, which then produces progesterone to develop the uterine muscles and cause them to thicken, and that's where we get our slippery cervical mucus and the likes. So we'll go with A still, but that is a lot of added information to what we already knew. So thank you. Is everyone okay with that?
Okay,
Please.
okay, so the temperature, the increase in body temperature, yes, it's alluded to, like it's because of the progesterone rise, yeah. But during ovulation, as Hiral said, like the progesterone doesn't increase that much per se. Like after ovulation, when the egg ruptures, that's like it transforms into corpus luteum. That's what is that's the corpus luteum is what starts producing the progesterone. So a rise in temperature is yes, right, due to progesterone, but literally the progesterone during ovulation is not that high. It usually goes up immediately after ovulation, after it has been released through the corpus luteum. So in this question, like the most correct answer will be rise in body temperature, yes, because through elimination, the other answers are wrong because there is no decrease in progesterone. Literally before ovulation, there was no progesterone, like the progesterone levels were low. And if it says fall in LH secretion, that means LH usually goes up during ovulation, and there's no decrease in the body level of estrogen. Estrogen is a bit still high during ovulation. So we'll go with A. I don't know if that makes sense.
Yeah, it makes a lot of sense.
Yeah.
Thank you.
Yeah, A is the best answer. A is the best answer.
So thanks a lot for that, Fatush, Dr. So let's go to number 13. If we could please go down. So a lack of or decrease in insulin hormone receptors on cells can result in a lack or decrease in insulin hormone receptors on cells can result in type C, choice C, type 2 diabetes mellitus. Remember that type 1 is usually caused by a decrease in insulin secretion, while type 2, we have a resistance. Our cells become resistant to the insulin, so they don't do their function. So a lack or decrease in insulin hormone receptors. So we have less receptors, so the cells are resistant to insulin. This is type 4 hypersensitivity, or the cell mediated, which are activated by the T lymphocytes. No, sorry, that's DM1. Sindio? Sorry, yeah. So type 4 cell mediated hypersensitivity is mainly diabetes mellitus 1. Sorry, so higher. So number 14. So maybe someone can share about juvenile diabetes and another about diabetic insipidus. So diabetic insipidus, we have an abnormality in the release of what? Someone, I think I shared it just the other day.
We're having
vasopressin. Thank you, Rami. Yeah, or the
for,
ADH.
for juvenile, it's exact same thing as type 1 DM, just another word for type 1 DM.
Yeah, so the child is releasing a little insulin. Cindy, we have a decrease in the insulin release.
Yeah.
Is that due to the features haven't fully developed?
No, it's the autoimmune.
Okay.
Yeah,
So,
autoimmune, yeah.
So thank you. Yeah. So yeah, as Jesse Munga has just shared, Dr. Jesse Munga, type 1 DM is the autoimmune destruction of the beta isolate cells present in the pancreas. So thank you. So I can move to number 14. Which of the following cells, or Rami wants us to share about SIADH. Can someone provide us with a bit more information? When we have the inappropriate release of the ADH or vasopressin.
Now that one was just SIADH is just the opposite of diabetes insipidus. In insipidus, we are not having enough ADH, but in SIADH, we are having too much ADH. Yeah, so I think it's just someone wrote it as an opposite to the diabetes insipidus. Yeah.
Yeah, it's because he put the question mark. So but it's been shared on the Stanley has also shared. Remember, we have both central or neurogenic diabetes insipidus, which is, you can just it's central, so the CNS. So now we either have a problem with our hypothalamus or pituitary, and it can either be congenital or acquired, or we can have nephrogenic diabetes insipidus. And now that's where we have a problem in the kidneys, and it can also be congenital or acquired. And it can also be primary polydipsia, which now is associated with stuff like excessive water intake, just psychogenic illnesses. So nephrogenic or central? So nephrogenic nephrons, you can just tell. So which of the following cells are important phagocytes that eventually become macrophages? I think, here the answer is just obvious. It's C. The person who had answered had said lymphocytes. Remember, when we talk about lymphocytes, now we're talking about our B lymphocytes. You just expect them to release substances rather than become macrophages that engulf or participate in phagocytosis. So, which of the following clotting factors is an important link between intrinsic and extrinsic pathway and is deficient in hemophilia B? Not very sure. Maybe someone can share. Factor 9. Dr. Divya, would you allude to sharing with us in case you have any further information. All I know is hemophilia A is 8 and 9. Okay. So
No, she's saying hemophilia A is 8 and
8.
hemophilia B is 9. Yeah.
Uh, but that's that's a lot of information. Don't even laugh. Like now we know A comes before B, so A is 8 and B is 9. So thank you. That's so. Anyone else before we move forward? Treatment of hemophilia B. Yes, more press. What are you saying?
Yeah, I think that's for hemophilia A, desmopressin.
Oh,
Yeah.
So personally, maybe I'll look for cheat sheet for the intrinsic and extrinsic, so I'll share it later. So we can move to the next question. That's okay with everyone. So which of the following blood from the lungs enters the heart through there. So from the lungs, that's oxygenated, so our left atrium. Is everyone okay with that? So
Yeah, just a request again. I know I've said this a million times, but what are the names of the valves again? The 4 valves in the heart.
So we have the bicuspid, the tricuspid. I think tricuspid is in the left and bi is in the right, if I'm not wrong. And then we have the aorta and the pulmonary. Another name for, I think mitral is also the tricuspid, if I'm not wrong.
I think tricuspid is on the right because tri. That's how
Mitral is bicuspid.
Yeah, tricuspid right, bicuspid left. Yeah, because these things they can confuse you, but thank you, Divya. Yeah, nice. Mitral, also known as bicuspid. Nice. Me, I'm good.
So and then now we have the aorta and the it's the
pulmonary.
Yeah.
Yeah. And these are the ones that are also known as the semilunar valves or atrioventricular. I think these are the semilunar. Ama? Yeah, these are the semilunar. Atrioventricular is the bicuspid and the tricuspid.
Yeah.
So semilunar is hapo kwa exit. Okay, nice.
So atrioventricular. So higher. The T wave on the ECG represents I think that's the ventricular depolarization repolarization, because it's just at the beginning there. PQRST. Oh, at the end, sorry. Yeah. Maybe we can go through the ECG chart just quickly again. Does anyone want to share?
Okay, in the ECG,
Uh-huh.
the P wave represents the atrial depolarization. Then the QRS represents both the ventricular depolarization and the atrial repolarization, according to what I know. Then the T wave represents the ventricular repolarization.
Ebu, just go again, please.
The P wave
Uh-huh.
represents the atrial depolarization.
Uh-huh.
Then QRS,
Mm-hmm.
ventricular depolarization and atrial repolarization.
So
Then the T wave, ventricular repolarization.
yeah. So
Then which one is the one that is affected by hyper or hypokalemia between PQRS or T?
It's the T wave.
T for
Yeah. Yeah.
Okay, thanks.
Because I think it denotes high blood potassium levels when there is high peak of T wave. If I'm not wrong, that's what you say.
Yeah. When it's high, it will be peaked. When it's low, it will be depressed.
Yeah.
Yeah.
Yeah. So I think just
Please repeat that, sorry.
yeah, so we've said we have the PQRST wave. Sindio. So think of it this way: the P wave, first we have the atrium, which is the first to be depolarized. So the P wave represents atrial depolarization. Higher. And then next we have the QRS. So that represents the ventricular depolarization and the atrial repolarization. And then finally, we have the T wave, which represents the ventricular repolarization. So just think, first you have to have depolarization before you have repolarization. So your atrium will first be depolarized at the P wave, and then during the QRS, you'll have both atrial repolarization and ventricular depolarization. So now your ventricles are becoming depolarized. And then lastly, now you want your ventricles to be repolarized because they're the ones which were left out. So that's the T wave, which is affected by hyperkalemia. So as Gas has said, I think during hyperkalemia, your T wave will peak. Sindio, it should
Yeah.
peak. Yeah, because it's becoming depolarized now. So yeah, and then during hypokalemia, obviously now you expect it to be down higher. So, okay, so
And then when there is an inverted T wave, it
uh-huh.
denotes that there is a myocardial infarction.
MI.
Because there's sufficient, like when the T wave is inverted and then it's not like the real T wave, how it's supposed to be in the ECG.
Yeah.
Since the T wave denotes that it's the whole ventricular polarization, so it means that there's insufficient blood in the muscles of the heart. So the T wave is like a signal for both hyperkalemia and for myocardial infarction. From the ECG, you can tell that.
Yeah, which makes a lot of sense because obviously you'd expect your ventricles to have problems when you're having problems with issues with pumping out of blood. Sindio? So
yes.
Yeah, so I hope I can go to the next question. In case anyone hasn't had anything or has a question, just go right away. Okay, hopefully we can try and get to 30 questions. At least I think we're moving at about 15 questions per hour, which is just well and good. So renin is produced by that's the juxtaglomerular cells. Remember the RAS system, we have renin released. This renin goes down to the liver and cleaves the angiotensinogen, forming angiotensin 1, which is then converted to angiotensin 2 by the ACE, angiotensin converting enzyme. And this then causes the release of aldosterone, ADH, noradrenaline, and the vasoconstriction of the arterioles. And then from there, now you have aldosterone, which causes sodium reabsorption, which is followed by water, and also an increase in the amount of potassium which is released. Remember, from the renin is usually first is a negative feedback. 17 is B, not D. Oh yeah, sorry. Shrina, yeah, sorry, I didn't look at that. Yeah, 17 is B. So we say the T wave, ventricular repolarization. So we had just left the ventricles. Those ones were the ones which hadn't repolarized yet. So renin, there are a couple of things which might cause renin to be released. Remember, we have the baroreceptors present in the afferent arterioles. We have, if we have decreased sodium in our renal tubules, and just generally decreased blood pressure present in the body. So we can go to the next question if that's fine. So which of the following is produced by the Sertoli cells? The tumors in the kidney, I think, will also increase. Yeah, that's true. That's true. So thank you, Hilal. So which of the following is produced by the Sertoli cells? FSH, LH, just remember, most times when you have like this, FSH, LH, ACTH, all of them are coming all the way from the hypothalamus, so that's wrong. Testosterone, androgen binding protein, and mucus. Who thinks what? Androgen binding protein from Hiral. Hiral, maybe you can tell us. Testosterone is released by?
The Leydig cells, I think.
What is there?
The Leydig, let me type.
Yeah, the Leydig cells. Who has additional content? To that. So we've spoken about Sertoli. Yeah, I think, yeah, remember prostate gland, that one is just for the basic fluid present in semen, which neutralizes the vaginal acidity. So number 20, if that's fine by everyone, please. So number 20, during ovulation, all of the following occur except rupture of the Graafian follicle, estrogen production is very low, FSH and LH production is high, formation of the copious luteum. Personally, I'd go with formation of the copious luteum, just because that happens after ovulation, but I'm probably wrong. Someone please tell me what you think.
It's B because we said estrogen is high during ovulation.
Oh, it's very low. Sorry. I was a bit confused. Sorry about that. Yeah, I agree. Oh, sorry, yeah, FSH and LH all the way from our anterior pituitary. So are we okay with that? So choice B, that's what we've gone for. all of the following are components of innate immunity except monocytes, macrophages, neutrophils, and T lymphocytes. So remember, these T lymphocytes, essentially, like our lymphocytes end up releasing, let's just say content. And this content has to be specific to whatever we are fighting to, to avoid damage to any cells. Think about it. Macrophages will only engulf foreign cells. But for these T lymphocytes, they're producing something. So whatever they're producing has to always be specific to whatever they're fighting. So there's no way they can be a part of the innate immunity. So the answer is D. Man, I know like my explanations are not the best, but I hope they're just fine. Like everyone is understanding whatever I'm saying. So antibodies are produced by dendritic cells and Langerhans. So Langerhans, we say they're in the skin, and dendritic cells are present in the brain and they carry out phagocytosis. So T lymphocytes is our answer here. Thank you, Rami. Dr. Rami, B. I'd also go for the same.
How is it B?
What do you mean?
I think it's the best answer. Plasma cells will be the bestest of best, but since we don't have plasma cells, B lymphocyte would be the best, second best.
Okay.
Yeah, because all the others, they are not involved.
Okay.
Yeah.
Thank you.
Yeah, so here we're going with A, Sindio, because B lymphocytes are the ones which will produce our antibodies. Sindio? How come?
Yeah.
Because T lymphocytes mainly work via sending signals, if they are helper cells or if they're cytotoxic, then we just through the cytokines so B lymphocytes are the ones which have the antibodies. Is there anything to add there? So if no one has anything to add, I'll go to number 23. IgE is mainly found in blood, surface of muscles, skin, surface, or aqueous humor. I'll go with B. And that's because, remember when we were talking about type 1 hypersensitivity, we were talking about IgE. And that's where we have our asthma, our allergies, anaphylaxis. And just remember, like whenever we're talking about this stuff, we always talk about the mast cells and the role that they have to play in causing these hypersensitivity reactions. So we can go to the next one. Does someone have something to add? So which of the following antibodies is a pentamer? I think that's IgM. Yeah. Choice, boy. I think IgA is usually a dimer, if I'm not wrong. If anyone has the structure of the others.
Yeah, IgG is a dimer, IgD is a monomer, and IgA is a dimer also.
So
Willie, can I
According
just
to the
add
picture
something?
Yeah, please share.
that is
Okay.
on the screen, I'm seeing that IgG is a monomer, not a dimer.
Yeah,
So
but
maybe
it has the ability to form dimers. Sorry. Yeah, it's a monomer. So IgM we said is a pentamer. IgG is a monomer which can form dimers. IgD and IgA are monomers. I mean dimers, sorry. IgD and IgA are dimers. Sindio.
IG is also a monomer.
IgE is a
A.
monomer.
A.
Oh, yeah,
Yeah.
just sawa.
Monomer and dimer.
Oh, sawa. So
Willie, allow me to add something that Stan
please.
had said in one of the discussions. He was like, in order for you to know which one is an IgM and a pentamer, rather which one is a pentamer, it has 5 points. So for like IgM, you can just draw it out and it has like 1, 2, 3, 4, and 5. So that's the easiest way to remember which one is a pentamer from these options.
Yeah, that's actually very smart. Thank you, Divya. Yeah, so basically she's saying when you look at the M, it has 5 points. Rami, yeah, I'm just explaining. So when you draw the letter M, it has 5 sharp points. So where's the down pen on this? Does someone click white? What do people click when they but anyway, yeah, there we go. Thank you, Shrina. Yeah, perfect. So there goes Dr. Shrina for us. Thank you. God bless you. So all of the following are live attenuated vaccines. I hope we're together, Rami, Dr. Rami. I hope you were able to see the M on the screen. So perfect. Thank you. So all of the following are live attenuated vaccines except the tetanus vaccine. So I think that one we've looked at it so many times, probably even after here. I'll go and open those notes again. So a complement system is involved in opsonizing and lysing organisms. Release histamine is composed of immunoglobulins, is only found in children. I think I'd go with choice A. What do you guys think?
Yes, A is fine.
So perfect. So yeah, if there's anyone who wants to add anything, please feel free.
Just a concern. Shrina, can you scroll down? Yeah, I think we've done this paper. Yeah, we've done this paper.
Yeah.
Like, yeah, we have done this exact paper. If someone can also remember, like us, we spend a lot of time discussing 7. Yeah, even question 29 where Dr. Karemi added on the T regulatory cells. Yeah, even Weffler's syndrome, we corrected it to Loeffler's. Yeah, we've done this paper.
It took us almost 30 questions to. I hope that's not the tension.
That's why we were moving fast.
Did we finish it?
No, I think the paper we did, it was like we did this paper, but it was half. So I think the top parts that we did now are not part of it. Yeah, if you can remember. Yeah.
True, because we hadn't looked at fluids at all at any point.
So should we continue or where do we start from?
I think because we only have 30 minutes left.
But I don't think we had finished the whole paper because there's some things I've not seen. Or is it just me?
Maybe we can see the pages.
I don't think we've seen this part.
Like that.
Yeah, we've not
Okay,
seen
we
this
start
part.
from down going upwards. Okay, when we feel like, okay, we've done this, we'll stop.
Yeah.
From
So-
99.
So, is there a question below? There's nothing below.
No.
So, what amount of digoxin is contained in 100 mg of the following powder mixture? So, digoxin 1 and lactose 900 mg and digoxin 100 mg. So that's a ratio of 1:9. Sindio? So digoxin takes 1 and lactose takes 9. Our ratio of 1:9. So 1 over the sum of the ratio to find digoxin. 1 over the sum of the ratio, which is 10, times our total 100 mg should give us the answer, which is 10 mg. I'll go again. Digoxin to lactose is a ratio of 1:9. So from that ratio, we can then find from 100 mg, we have 1/10. So we add the ratios to get the total 10 times 100. Yeah, thank you. Thank you, Divya. Thank you, Shrina, for sharing it on the paper. So what volume of normal saline 0.9 weight by volume can be made from 4.5% grams sodium chloride? What volume of normal saline 0.9% weight by volume? Honestly, I'm not very sure how to do this question. Maybe someone can. But I think this is 500 ml. Just please share. But basically, the 4.5 is also weight by volume. 4.5 mg. Yeah, 4.5 mg. If 4.5 mg is in 100 ml, what about 0.9? Let me see. 0.9 times 100 times 4 by 4.5. No. Well, someone shared it in the group. Oh, awesome.
Is it like this? 0.9% is 100 ml. So what about 4.5%?
Yeah. That's so sick of me.
You don't convert the 4.5?
No, because
Oh, okay, fine. Okay, I've got it.
So from the way I've understood it, basically you already have, when they say 0.9 weight by volume, you already have your weight and you already have your volume. So for the 4.5%, that's where you haven't been given any volume present. So that's your unknown, obviously. So basically just 0.9% is equals to 100. What about 4.5?
Yeah, it means 0.9% weight by volume. It means 0.9 grams is in 100 ml of volume of sodium chloride solution.
Yeah. So thank you. Thank you, Shafi, Dr. Shafi, for that. So If I may, I can move to the next question. I just released, oh, sorry. What percentage is produced when 150 mg of powder is made up to 25 grams with a diluent? What percentage is produced when 150 mg of a powder is made up to 25 grams with a diluent? Percentage is produced from 50 mg. Yeah, 25,000. 100. What about 150? 150 times 100 divided by 25,000. Yeah, so what percentage is produced when 150 mg of a powder is made up to 25 grams with a diluent? So basically, if 25,000 mg or 25 grams is 100%, what about 150 mg? So yeah, I think that's self-explanatory. So 150 times 100 divided by 25,000, you get 0.6. If 150 mg, yeah, okay, perfect, yeah. So 300 parts per million as a percentage. So 300 over a million times 100. So ppm parts per million. So 1, 2, 3, 1, 2, 3 times 100. You get 0.03. The answer is dog. Yeah, thank you, Divya. So it's there on the group. Just remember parts per million. So per million, you always divide by a million. After per oral administration, drugs are generally best absorbed from the not too sure. Personally, I'd say, is it the stomach? I don't know. Someone please share. What do you guys think? The duodenum. So when you take drugs through the mouth, they are best absorbed in the duodenum. Yeah, I think we
something on number 95 because I don't know if it was in a previous paper, but we had seen a similar question. And so the greatest site for absorption of drugs is the duodenum and the jejunum.
Yeah.
And now when it comes, if there was an answer or rather a choice such as jejunum, that would be the most correct.
Yeah, I think we're studying, it was proteins. We're mainly looking at protein absorption, that question, if I'm not wrong. Sindio? Even just, I think there was a question also like that, and we also said the same thing. Yeah, for food, as Corazon is asking. So we said, because I remember someone was arguing, the duodenum is such a small part, and the jejunum is much larger. So, and then even when we looked, we found because of the size of the jejunum, it's more likely to, for food absorption to occur in the jejunum. But if you have only duodenum and ileum, duodenum is the answer we go for. Sindio? And then the ileum, we said mainly for vitamins and lipids.
Then there's a question on the chat box.
Yeah, that's what we're saying. So we're saying for both food and, as Dr. Gas is saying, drugs, jejunum, and for even as you've said, Jambi, Dr. Jambi, about food. So yeah, same thing. So the answers are just the same from what we found previously.
No, I think from the previous discussion, for drugs, the drugs are mostly absorbed in the duodenum, and the food is mostly absorbed in the jejunum. So if you are being asked about drugs, the highest percentage is absorbed in the duodenum. If you asked about food, the highest percentage is absorbed from the jejunum. There is that difference.
So, Dr. Gas, are you okay with that?
I just request for just the reference, because there were a lot of references saying the opposite. So kindly just share the reference for that, especially for drugs. For food, I have no quarrel. For drugs, saying that the duodenum is the major one as compared to the jejunum.
It's okay, I'll share.
Thank you.
So, the screen has disappeared. Sorry. So, higher. Number 94. Dose dumping is a problem. Ileum-based absorption for vitamins and lipids. Jejunum for food. Duodenum-based absorption for drugs. So we said we'll look more into drugs. It might be the jejunum or the duodenum, but we'll share further. So dose dumping is a problem associated with one of the following dosage forms: compressed tablets, modified release tablets, hard gelatin tablets, soft gelatin tablets. So here the answer was modified release tablets, but I know the gelatin is hard to be released when it's not in the stomach. So that one I understand, but what about compressed? Oh yeah, compressed tablets, they have less disintegration, if I'm not wrong. So I think that's why B is the answer, but maybe someone can share a bit more on that before we go to the next question.
I think B is the answer because modified release tablets are designed to release the API for a prolonged period of time.
Yeah.
So when they release the API at once, that is what we refer to dose dumping, and that's why it can be a problem for the modified release tablets. But for compressed tablets are meant to just release the dosage form at once. Once they disintegrate and dissolve, then they just release it at once.
Yeah, I think for the capsules, it's a bit hard for them to just, open up when they're not in the body, and also for the compressed tablets, like the disintegration when they are not in the body is also a bit hard. So I think the modified tablets are the ones which, regardless of wherever they are, they'll still release the contents of their materials. If I'm getting you right. So, the next question. The following factors affect the effectiveness. I think moisture can affect for gelatin tablets. Yeah, you're right, Trina. But I think because then again, they're capsules and everything. I think the answer, which, like the most significant answer for the question is B. So perfect. So the following factors affect the effectiveness of a preservative, except obviously concentration, pH, the oil-water partition coefficient, but not surfactants below critical micelle concentration. So the critical micelle concentration is, like above the critical micelle concentration, then micelles will form. So if they're below, then now we have no micelles forming. So that doesn't really play a role in determining how effective our preservative is. So just basically, we'd prefer if our CMC was lower, so that we don't have, micelles present in our drugs. If everyone is okay with that, we can.
Pardon on that last point about micelle concentration.
So saying the critical micelle concentration, below that, so above it, we have micelles forming.
Yeah.
Yeah, and below it, we have no micelles. So just generally, we'd want a low CMC. Sindio, surfactants below critical. Yeah, just generally, we'd want a low CMC in our drugs to prevent the formation of micelles within the drugs themselves, if I'm not wrong. Sindio? So
my question
really?
is for CMC, like, don't we, when we've exceeded the CMCs, that is where now mixing will occur. Because you're saying we don't want to exceed the CMC, but I thought CMC we need to kind of exceed it for mixing to occur. I stand corrected. I don't know.
I was thinking, the way I was thinking about it, I was saying in terms of when we have the surfactant. So do we want the surfactant itself to have a high CMC? Or do we want the mixture of our product, when it's in the body, to then have a high CMC? Am I making sense?
Oh, I've gotten you. Sawa.
Thank you.
There was someone who also wanted to say something.
Yeah, let me just read the chat box. So back to dose dumping, let me just add what has been shared. Dose dumping is a phenomenon in which relatively large amounts of drug in a controlled release formulation is quickly released and potentially toxic quantities of the drug is introduced into systemic circulation. So just generally, when we talk about dose dumping, we are talking about controlled release formulations, which is almost similar to modified release tablets. So additionally, dose dumping proves to be fatal in the case of potent drugs, which might have a narrow therapeutic index. Higher. Can we talk about the concept of CMC? That is from Dr. Brenda. Yes, we can. So basically, the CMC is the concentration of surfactants above which micelles form, and all additional surfactants added to the system will form micelles. So that's what I found online. And yeah, maybe someone can add something on that. So basically, I was saying we want the surfactant itself to have a low CMC so that we don't have surfactant micelles present within our drug, you know. Yeah. If anyone has anything to add on that, please.
Just perhaps just something to add, something small on the same, that it is a concentration above which if you add any more surfactants, micelles will form. Below the CMC or at the CMC, the surfactant is just at the border between the oil and the water. So when you add more surfactants is when you start, the mixture will now start mixing, because it will mix in that form of micelles. Yeah. So that's all I had to add. I'll send an image that really you can follow with that explanation.
So, thank you, Dr. Gas. So so something has been shared on the group. We can look at that as we proceed. Yeah, there's a lot to catch up on, Adiata. So which equation is used to predict the stability of a drug product at room temperature from experiments at accelerated temperatures? What do you guys think?
I think C. Yeah, C. Oh, so I wrote C. C.
Oh, so stability of a drug product. Can you explain? See, Arrhenius ni zile ma Arrhenius acids and Arrhenius bases, if I'm not wrong.
Yeah, there's that. But there's also this Arrhenius equation. I think it's A will equals E, something R to the power, or not powers. Shrina can write it, but it's usually now activation energy of the gas constant multiplied by temperature. So it is, yeah, so it relates those things. Yeah. So
for the Stokes equation, we know that has stuff to do with viscosity. For the Michaelis-Menten, that has stuff to do with enzymatic rates for different substrates. Maybe someone knows something about the Young equation. So that has matters to do with surface tension. So we said Stokes' law, just remember, that's viscosity. The Young law, that one has matters to do with, you know, just surface tension and equilibrium forces in regards to that. Arrhenius, we've said that's the equation which is being shared by Shrina. Yeah, and the Michaelis-Menten, that's a matter to do with enzymatic rates in regards to substrates. So I can move to the next question if that's fine. In flocculated suspensions, particles form loose aggregates. Rate of sedimentation is low as each particle settles separately. A very close packed sediment is formed, and suspended material remains suspended for a relatively long time. I think the other day, Dr. Noor was sharing with us about this. Maybe if you can, could you please unmute and share? I remember you talked about deflocculated and flocculated. Please. Just repeat what you said. If you're with us. Dr. Fatma Noor. Or anyone else, please don't.
I had shared the photo. I didn't like technically explain it, but I had shared the difference as a photo in the group.
Yeah, but can you just share with us why A is the answer? Or cause I think I remembered something to do with deflocculated had a higher rate of sedimentation, if I wasn't wrong. I don't know. So I think gas was also somewhere in the fold. Maybe he can also come out and shed some light.
In a simpler language, flocculated have large particles. Deflocculated have very small particles. So the flocculated, which has big particles, settles very fast and forms loose aggregates, which upon shaking, they can easily disperse again. But deflocculated, since they are very small, they settle slowly and they form, what do we call it? A cake, which when you shake, it does not redistribute again.
Oh, that's an amazing description. So I hope everyone got that. When we say flocculated, we are talking about large particles and they'll settle down quickly. Deflocculated, they're small, and when shaking, they form a cake and they don't form a suspension again. I'm out of it.
Yeah.
So, all right. Number 90, if that's fine with everyone. So we expect the cake to be formed with the deflocculated, and they have firm aggregates because they're small in size. So a satisfactory. So just back to that. So we expect the flocculated suspensions to sediment faster. So answer for questions 92. So question 92, the answer is C. And we said when you hear Stokes' equation, you think about the viscosity, matters to do with viscosity. Arrhenius equation, that has matters to do with stability of the drug, and that's the equation there on the right. Michaelis-Menten, that one we said it has matters to do with enzyme stability, enzyme rates on substitutes, and for the Young's, I hadn't even written it, Young equation. And for Young equation, we said it has matters to do with surface tension. Surface tension.
I think someone had shared and said that the Young deals with infant dosing. I don't know.
So
that was Hopkins.
For dosing, it's the Young's formula, not the Young's equation. For calculating dosages, it's the Young formula. Young's formula. Yeah.
That's all. Thank you for that. So you're welcome. Okay. So a satisfactory suppository base must meet all of the following criteria except should have a narrow melting range, should be inert, should dissolve or disintegrate rapidly in the body cavity, should melt below 30 degrees Celsius. So I think it should have a narrow melting range, which is correct. Should be inert. That's clear. Because you don't want something acidic up there. Should dissolve or disintegrate rapidly in the body cavity. That's true. And should melt below 30 degrees Celsius. I think if that was the case, then we want something which will, melt within the body temperature ranges. So I think D is our answer. So number 89. think this can be our last question, and then we'll come back. So we know the body temperature, we expect it to be at about 37.2 normally, but the ranges are from 36.5 to 37.5 degrees Celsius. So, which of the following definitions? I hope you got that. So, can we discuss on suppositories quickly? Definitely, Dr. Gas. Please tell us where to start, and then we shall follow. So, what exactly do you want us to talk about?
Oh, just okay, just an overview, kindly, like the bases, stuff like the bases. Yeah, so just an overview. I'm also not that well versed with suppositories, hence my quest.
So just generally, we know that we can either have, for a suppository basis, we can either have our lipophilic bases, or we can have the hydrophilic bases. So, and we can also have the synthetic bases. The lipophilic bases, those ones are the ones melted at body temperatures, while the hydrophilic are dissolved in rectal fluid. So gelatin and PEG, polyethylene glycol, those ones are hydrophilic, so they dissolve in rectal fluid, while the lipophilic bases are like cocoa butter and hard fats, and our synthetic bases are marsupial, etepsol, and masa estarium. Let me see if I can look for something else. In case anyone has something they can add, please join in. Dr. Gas, is there something in specific?
Yeah, Nura sent something nice. I'm good. Asante.
So, something shared in the group. No, it's nothing. So, okay, we're good with that. Okay, number 89. So basically, we've just said we have the hydrophilic and the lipophilic. Lipophilic, we don't expect them to dissolve in rectal fluids, so they'll dissolve or due to body temperature. Those are cocoa fats, just generally fats. And then hydrophilic, now that's where we have our PEG and our, we said PEG and what? Polyethylene or gelatin. Yeah, so those ones will dissolve. So, which of the following definitions of the common tablet processing problems is true? Capping, we have partial or complete separation of the top or bottom crown from the main body of the tablet. So capping, yeah, basically just the cap, So bottom or top crown, so that's true. Picking, adhesion of a tablet material to a D-wall. I mean, that's if someone has the exact definitions of this, I think there was somewhere in the group, if I'm not wrong. So we can finish up with this one and then we call it a 1 hour break. Sindo, before, or 2 hours, sorry, before the next session.
Yeah, I think Stanley had sent a nice image on the same, having all those defects.
Yeah.
Yeah.
Don't know why I'm not finding it, but I'll look for it and then share it again on the group if that's fine. So can we call it a 2 hour break from now till 8?
Yeah, we can call it.
So thank you, doctors. I plan on seeing you guys soon. Hopefully someone else will take the mantle and probably do a much better job than me. So I will see you guys in 2 hours. Wait, guys.